Well, hi, everybody. You're here at Cyberhuman Frontiers with us again. Why do we call this channel and these shows all about Cyberhuman Frontiers? Because that's what's going on. That's where we're going. We've been discussing things at the genetic level for a good while now, and there's a reason for that. That is like foundational. And what's going on is further and further and deeper research into how to use biological molecules and merge that together with computer technology so that designed molecules can be integrated into life forms. Now, it can be done in two ways. One would be for building that molecule so that it would be an interface device to things, to other computer equipment or other peripherals on the outside of the organism, or, if you will, the human. And the other would be to bring these designed molecules on board and have them integrate so that it changes the operational control of life processes in cells. And there's a little bit of both. There's a distinction between the two. One approach is to be able to develop better input-output into an organism or a person. And the other one is to literally redesign the internal functioning of the organism. And in a kind of a standalone way, once that new developed and written genetic software code is brought into that organism, it becomes something different. Or, as I said, a little bit of both. So before we really get going on it, we just want to get our community guidelines disclaimer out of the way, and bear with me as I try to read that as quickly as I possibly can. The points of view and purpose of this video is not to induce fear, incite violence, nor harass, nor bully in any way, but to share and exchange thoughts with other like-minded individuals curious about the subject. This is to encourage conversation and awareness. Conversation is good. That can happen right now. And awareness is something that will sink in over time as you ruminate and ponder over the things that we show you here. You'll start to see other aspects that tie into life here and now today as you see it. The opinions expressed in this video belong solely to the persons espousing them and are not a representation of the channel Cyberhuman Frontiers. None of our videos claim to give medical, legal, political, or financial advice. This content is created entirely for your entertainment. Our channel and our shows from time to time may contain certain copyrighted works not specifically authorized, but which we believe in good faith are protected by federal law and the fair use doctrine for one or more of the reasons noted in the Copyright Act of 1976. We make it a very standard and thankful practice to include the links to other content creators that we feature in our shows, and we really do appreciate them. Many fine and brilliant people, and you'll see a few tonight. And those links can always be found in our show notes, usually either during, before, or definitely at the conclusion of a video. So thank you for that. So as we get started here, TMC, how are you tonight, sir?
Indy, I'm good. I'm going through some old notes and I'm finding some links that are taking me down some rabbit holes.
All right.
Once upon a time, we're pretty amazing. So I hope we get that far in the show. Yeah.
I've seen and sampled some of the previous work that you did, and if it's almost as if you've been a prophet with some of the content that you created even a year ago. You were looking at patents that were there, and then materials science development and progress, and it's almost as if you predicted a future that we now see come to fruition just a year.
Me.
What?
No way, man. It wasn't me. There are people have been predicting this for a long time. You could argue this was predicted biblically. You could argue that. And make a very good position for that this is biblical. But then, you get into the 50s, 60s, 70s when you start seeing material sciences and the mechanization and the automation and the computer science and software and all that start coming into its own and actually getting some legs. And then you can start seeing the actual nuts and bolts, the mechanical pieces of how all of this, revelatory kind of stuff is supposed to manifest, the machine, if you will. That's why I use the term nuts and bolts. But it's not me. I just happen to look in places that, stuck out, that seem to resonate.
Dude, there's a certain knack to knowing how to look in the right places.
Yeah.
Because in order to do that, you've got to sample something and then connect some dots in your own mind and say, this is a further push toward the edge of what I already know, and and this thing that I just ran across ties into that. So let's see where that goes, and the picture starts becoming more filled out with the facts. And you've done a really, pretty good job of that. I'll just say pretty good, but I think
enough on all that. Let's go with what you got with the videos, or let me still share. Yeah.
Oh,
good.
You want to do it right now, or do you want to?
Let me take it real quick.
All right, so I got this little thing here, and so what you're looking at is we're continuing on from where we were last week's episode. We did just graze on the topic of how to build nano devices, and of course those are built of nano-sized, molecular, engineered little particles. And various terminology that people use, and they're all sort of interchangeable, whether you call it nanoparticles, nanomolecules, nanotechnology. Nano means small, and it's all about that idea. So we have here a little picture of an artist's representation of what could be a future designed little device, and this would be extremely small. And if you take a look at it, there are a number of possible appendages which would be pre-built molecular structures which could be attached. So where my mouse pointer is over here on the right-hand side would be particular little attachments, all right, or appliances that could be connected to one of these arms on this thing, okay? And so you've got the main body of it. And if it has any sort of a central processing unit in it, it's going to be in the main body. And it is constructed of pieces of various molecules, but particularly it's going to have DNA in it, because that really tends to be a handy molecule to build structures out of, believe it or not. This is all stuff that we've featured in more detail in previous episodes. So we encourage you to check out some of the other ones. But so we have here possible appendages that can be designed and attached to a little DNA strand that's hanging off the main body of this little nano robot. Okay, so we have a clamp. All right, it's kind of like just a two-fingered thing. We have a claw. We have I can't even read this thing, a spatula. Okay, something to scoop things out. We have what the artist conception calls this a ray gun, but what that would really be is something that would be able to directionally and coherently shoot out a very tiny beam of either light or perhaps electricity or electromagnetic radiation. So that's a sender. The ray gun here, as they called it, is a sender. And then up here, we have a receiver, which they're calling a radar antenna. And if you know much about antennas, you take a look at the structure of this. We have a receiving point, and then we have a parabolic reflector, which concentrates whatever it was receiving from a directional source right to the heart of the receiver, right there. And so anyway, that's just an artist's conception. Now, on a device, on a nano-sized device like this, it would be completely possible to build molecules into it, which would be either senders or receivers of electromagnetic radiation, light, thermal or chemical sensitivity, etc. So just by way of extremely quick review, let me find the right one here. Let's see where, I think this is it. Right. by way of extremely quick review, last week we featured a presentation by this man who is, if he doesn't call himself an electrical engineer, he could be, because he spoke very fluently in electrical engineering terms about building biomolecules to do what is done in electronics with logic gates, etc. So that's in last week's episode, as well as the link both in last week's show notes as well as this week. This is Christopher Voigt. I found him to be a really, really smart guy, and it's not all that long a presentation. So if you want to review that just for fun, you can do that. So where we're going with this is why we're taking you on a journey in the learning and the awareness and the state of the art in what is going on with molecular engineering. And what is at the heart of all this molecular engineering? Well, it's code, it's software, and that's the holy grail of the whole thing. What happens when you want to build something and make it do what you want it to do? There have to be instructions. And DNA is an extremely compact and highly dense form of storage of code software. It is. And it's in all life forms on this planet and probably elsewhere too. Anyway, so that was what we featured there. So now as we go forward, if I were to just tantalize your thinking about what we could do, I'm going to feature another little one here with a couple of guys, and this is not really a very long presentation. I think that I won't necessarily need to play the whole thing, but you can watch the whole thing, 5 minutes long. This is from the American Chemical Society, and this is one example of what you could do if you wanted to build a molecule or an aggregate of molecules. We're going to review once again. We used the term, in our last week's show, we used the term molecule versus monomer. Well, monomers generally are a little bit larger than a molecule, and they're usually built of multiple molecules, all designed and linked together chemically for a purpose. So these guys decided that they would work on the project of creating molecules for sensing logic operations. And how would they do that? Well, they need some input-output. They need to get some way of communicating with the molecule, and they decided to do this by way of light. And so it's only going to be a couple minutes here, and this is just one more fascinating avenue. And I have to stress that there are so many different prongs to this field of research, all which finally come down to the foundational level of life and what's at the core of it, and all of which come down to utilizing or manipulating genetic code, that's that software, for the purpose of making these little designed molecules be where you want them to be, have the characteristics you want them to have, and then activate, do what you want them to do when you want them to do them. And so here we go. He's going to start off, believe it or not, with Lego blocks. And it's only a couple minutes long, so pay attention. Let me make sure I shared I think I shared the audio. TMC, if the audio doesn't come through right, just flag me. All right, here we go.
And believe it or not, the design of our systems starts with this. Just children's blocks. And as you can see here, there is a blue block which represents a lumophore, and there is a green block which represents a receptor.
Okay, hold it right there. The blue block representing a lumophore and the green block representing a receptor. This is the way engineering always starts off. We start simple, we start with theory, we start with building blocks, and then we try to integrate that into something real world, tangible. And anyway, there's his initial conception. And TMC, is this loud enough, by the way? He seems a little quiet. It's good? Okay.
It's coming through.
Okay.
Good.
Very good. So keep in mind the subject is these guys are about the task and process of developing a luminescent photo-induced electron transfer molecule. A lot of words. So it emits light, and you're gonna take a life an existing molecule of life and modify it. Here they go.
And they are held together by a red spacer. And as we can see, the blue block and the green block do not overlap, so that there is a substantial autonomy between the lumophore and the receptor.
Well, that's pretty simple, right? So you're going to have a base molecule, whatever it is, and there's no problem at all having you make the stretch between this being Lego blocks or something extremely, extremely small that you'd need a powerful microscope to be able to see. There's no problem at all. As long as we know what the characteristics are and the way to bind these things together, we can do this, and they've been doing it. Okay, now the young engineer takes over, and he describes exactly how they do it.
Hi, my name is Glenn Wright. What we have here is the molecular version of the building blocks that we just saw, in that we have a lumophore, a spacer, and a receptor. By designing our molecules in this way, we ensure that the individual properties of the original components are preserved. When we're designing our exact molecule, we must consider...
What he's referring to there is it's necessary to have some isolation between the lumophore and the receptor. If they were, in physical terms, if they were to be too close to each other, let me see if I can get this thing back to there. If they were to be too close to each other, they might contaminate the operation or the function of either one. So it's necessary isolation, but they're both connected together. And I'm sure that there'll be some input-output between the receptor and the lumophore, and it probably has to go through the base molecule, but it's under some sort of control. And now, when we're talking about control, we need to keep in mind this video that we featured last week from Christopher Voigt, who was showing the real down to the nuts and bolts of how you have that isolation and control within a molecular structure. So these guys go on.
The molecular version of the building blocks that we just saw, in that we have a lumophore, a spacer, and a receptor. By designing our molecules in this way, we ensure that the individual properties of the original components are preserved. When we're designing our exact molecule, we must consider two things. The first is PET thermodynamics, which is governed by the lumophore and the receptor, and the second is PET kinetics, as governed by the spacer. When light is excited onto such a molecule, it is the photo-induced electron transfer that prevents it from becoming luminescent, and so the compound remains dark.
When the target is added, green light flares up.
I'm Yue Hu. I am synthesizing a luminescent PD sensor here. One of my sensors has been purified by crystallization.
I'm Jack Hayden. I'm doing an electronic absorption spectrum of a PET sensor. This will allow us to get the optimum excitation.
PET sensor, photo-induced electron transfer. So if you guys just keep in mind, in the back of your mind, how do we end up being able to make something like this? Okay. The way we end up being able to make something like this is it starts off with the making all of the components that are going to make up the ultimate combined machine. I mean, think of this as a little machine that, you got to build it piece by piece. So that's what these guys are actually doing. Brilliant, brilliant people. Brilliant people. So I'll run a little bit. This is not a very long video. We'll just run a little bit more of it just for your entertainment and information here.
For luminescence.
I'm Ruxi Xu. I'm measuring the luminescence emission spectrum of the sensors that I've made. The luminescence intensity will be measured as the function of target concentration.
Here is an example of a luminescent PET sensor which went all the way all the way to a hospital or an ambulance near you. This little plastic cassette takes blood from a vein, whole untreated blood goes in here, and then the blood circulates around this little tube, and each of these little spots is a luminescent PET sensor molecule designed according to the principles we discussed. And these spots will emit luminescence, which is dependent on the concentration of sodium in your blood or potassium in your blood, for example. Here you see our previous 3-block system expanded to 5. In other words, we have 2 receptors looking for 2 targets, or
See how engineering works? And I'm going to reiterate back to, once again, I don't know if this thing will run, because I haven't reloaded it, but, oh gosh, where was it? Okay. So notice he expanded it from just 3 components up to 5. All right. And any one of these diagrams would tend to serve the purpose. But what he was talking about was having something that has, I got to just run this a little bit so that it refreshes itself.
And a NOR gate is...
Okay, there we go. So now we have 2 inputs and 1 output. Well, that is exactly what these guys are talking about. They've now added 2 inputs for 1 output. So notice people from, these guys may not even know each other, but from different and peripherally related fields with people who may not even know each other, they're all working in the same direction. Now here, depending on how this molecule is built, this thing with the 2 greenies is going to become either an AND gate, an OR gate, a NOT gate, or a NOR gate. All right. And this is a logic gate. Well, they're building this at nano-sized devices. And so what Christopher was talking about here is how you go ahead and build, and then you build up with more of these gates into something like this. Now, if you see this diagram here, he's got a whole lot of gates on here, and they're all of the various types of logic gates. So this is bringing it right from the perception of it. Forgive me, Chris, if you're not an electrical engineer, but I certainly would give you credit to either be or become a really good one. Extrapolating this all the way from electrical engineering, and this is the science of microprocessors. This is the science of logic gates. You just build it up a whole lot more and put a time base in there and some memory, and you've got a small microprocessor. Building it all the way up from the level that these guys are at up to this level. Absolutely fantastic. And when you do enough of this, then you finally get to the point of being able to build yourself one of these guys. So, and look at the caption there. Science fiction? No, not really science fiction. I want to take a second and just check out what people might be talking about out there in the chat. And okay, so we are on a new frontier. We believe it or not, TMC and I have a direction. We have in this exploration, we're bringing you along with us into a greater understanding of the physical world, what's going on with material science and engineering. And we have some other things that we have not really opened up yet, but they have to do with more of the quantum effects that we've actually seen operating in our world, and most likely are operating in living creatures now, and some of the other things. We're not getting there yet, but we, that's ultimately where this all goes, is further out into what now might be completely unfamiliar to you, but you're going to see experiments, you're going to see documentation, you're going to see patents, you're going to see designs, and you're even seeing it now with quantum computers to some extent. And we're going to tie this all together for you. So yeah, good to see you out there, Dynamics. And let's see what else we have here. Yeah, silicon electronics, absolutely. But we're moving from silicon-based electronics to more organic type living molecules.
It's going to be a hybridization.
Yeah.
We're going to go through a hybridization first, and then I think you're right. We're going to ultimately go to or get to a point where it's completely biologic, where we don't need the metallic pieces as much.
Not as much. That's right.
Silicon, so to speak.
Well, you know what? The metallic pieces, TMC, there will be the token metal atom here and there. There'll be the token electrically charged metallic ion somewhere, because you need those, and they're in normally occurring living creatures now. Right. There are metals in us, and they're there for a reason. They help with the supply or the soaking up of electrons, and that is kind of, it gets into complex stuff. I don't want to bury people in it, but electron transfer is very important when you're talking about having cellular processes operate, because the electrons change the charge differential between two adjacent atoms, and they make chemical reactions happen. So there's always electrons in there somewhere. So these guys got about a minute left. I think we're going to let this go. And once again, American Chemical Society, a PET molecule, photo-induced electron transfer molecule, luminescent. Okay, I'm going to finish it up.
Two chemical inputs. And the moment you have that, in this molecule we have Boolean and logic for the first time.
Isn't that amazing? Okay, people in these labs building these segments of this, and now for the first time they have manufactured a living molecule that works like a computer. And this could be designed in such a way as either to be an AND gate or an OR gate or a NOR gate or a NOT gate, any logic gate. And you could even have more than two inputs. You could have three, you could have four. And another logic gate function is an inverter. And an inverter would work something like this. Here's your input and your output. Let's say this was an AND, right? And so if you have this and this, then you get the output. But we can put an inverter at the end of that, so that under all normal conditions, you're getting the output on. But when you have input 1 plus input 2, activating it, then you get off coming out. It's called an inverter. This is all pretty basic stuff. You can look it up on the internet. There's infinite resources about computer gates out there. But for the purpose of this discussion, it is building things out of biomolecules that do what computers do. Here they go.
This logic is found almost everywhere, from the language that we use to the technology to the biochemical processes that keep us alive. It then makes sense that we should be able to design a molecule that does the same thing. The first example of this actually being done was in this lab in 1993. Computing molecules are now being used and applied in smart diagnostics. We can conclude then that luminescent PET molecules for sensing and logic will continue to grow well into the future.
There you go. The Marr family, Belfast City Council, I don't know who these people are, but they're thanking them, so I think I'll thank them too. And of course, while I'm at it, it never hurts. You can do this for us too. You can go, I have to sign in first, then I won't do it. So anyway, you can go right ahead and look this up to your heart's content. Hang on a second here. My thing got stuck. Okay. So foundation, this is groundwork. This is where it's at. Notice that he said it was in 1993 that they first did this. So that's kind of cool, huh? 93, 03, 2013. So I'm going to go back to, I am going to go, let me put the share back up here. Going to go back to that diagram that we had of the little robot-y thing. And it always starts theoretically, and then it starts simply. and then it gets more and more sophisticated. And then finally it gets to the point that it's good enough that you can actually integrate it into a living creature. So it may be science fiction, but you know, one of the mottos of our show is science fiction comes true. And my goodness, it does. And it is. So got a, let's see here. I have them here somewhere. Okay. This is going to be interesting. And I'm going to try to narrate this a little bit as we go along. This is part of a presentation which is in your show notes. This is a person who decided that he was going to make his career in computer animation of real stuff. And this is Drew Berry. You're looking at animations done by a man named Drew Berry. And so let me just run through this a little bit. It's not very long. This is only 1 minute and 37 seconds. So I'm going to kind of go slow and then move right along. And I'm sure by then TMC will, you're just going to want to get something off this at some point. So just jump in whenever you want. You're looking at DNA. You are looking right now at the double helix. Okay. And what is this? This is the backbone. The red thing is the backbone. It's made of sugar. Believe it or not, a sugar called ribose. And it even is a little more complex than that. It's called deoxyribose. But basically, in a word or three, it's a 5-carbon sugar. So the outer part of this spiral is a 5-carbon sugar. That's kind of what holds the whole thing together. And that's very repetitive. There isn't much that differentiates the part of it that's over here one side from what's on the other side. But now these yellow thingies inside are called base pairs. They are also called nucleotides. And this is where there is a specific code, because even though these all look the same, they are in very, very regularly occurring in all life. They are one of 4 types of nucleotide: cytosine, guanine, adenosine, and thymine, adenine and thymine. So I'm going to run this if I can, and you're going to see what some of the processes in a living creature are. We are at the subcellular scale right now. We would be inside the nucleus of a cell, and we would be into the chromosomes of a cell, and we would be down to one part of one chromosome in that cell's nucleus. Okay, there you go. Now, for the purposes of illustrating what this is, this was just unfolded. Now you can see what this would look like if it was uncoiled, so to speak. All right, now we're going to go further. Oop, now something else happens. Now, why is this significant? Because these nucleotides, if you remember I mentioned them, every one of the ones on the bottom is the mating complementary partner of the one up above. And they are cytosine, guanine, adenine, thymine. Always those 4. Okay. Now, you might ask why this thing separated? Because this is what happens in a cell. When your cell is going to want to make some sort of a protein or an enzyme or any other structure, it needs to get the code for how to make it off of this stuff, your DNA. And this gets a particular section of this is going to get copied, and that will produce a given protein or enzyme or other molecule. Okay. I just might play this again with him narrating it, but in the interest of time, I try to go faster. So he's showing the mirror image aspect of this, and this is his great work in computer animation. Okay, now they're zipping back up together again. Now they're coiling back up. So what happens when DNA is going to need to be used is it goes through that process of uncoiling and unzipping. And one of the unzipped parts goes through another cellular structure called a ribosome. And a ribosome, big long word for something that is, you know what it is? It's a copy machine. That's all it is. It's just a copy machine. So that unzipped thing gets run through the copy machine, and out the other end of the ribosome comes a copy of it. It's that simple. Now what's he going to talk about? Okay. All right. So what you're seeing here is a ribosome. You are seeing DNA that's coiled up, both parts of it together. They're going in one end of that ribosome, and notice a single strand coming out. All right, let's let this go a little bit further. It's a very short clip here. So this thing is the ribosome. It is splitting that DNA into one or another part. Now over here, we have something else going on, which is the other part of the copy machine. I think I'm going to go ahead and let him do his, I'm going to go ahead and let him narrate this. He'll do a better job of it than me. But let me let this finish up before I play his thing. It's got 10 seconds left. This is something that's going on in every one of the cells of our body, millions of times a second. Every cell is doing this, synthesizing, replicating, or dividing. And there we go. That was that one. But, let me go ahead and find this thing here. Okay. Drew Berry did some great stuff. And even if I play this whole thing, it still isn't very long, so you'll kind of want to pay attention if you're interested. I know I'm pretty interested. Understanding of how these mechanisms work is a big key to being able to engineer things to either imitate or augment those functions, or edit DNA, all of the above. Edit, add, subtract, delete, splice, all that stuff. Okay, so here he goes.
Let's begin. This is DNA in its classic double helix form, and it's from X-ray crystallography, so it's an accurate model of DNA. If we unwind the double helix and unzip the two strands, you see these things that look like teeth. Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right.
You're on mute.
Thank you. So if I didn't thank you, TMC, I forget to unmute myself sometimes. If I gave a quiz question and he said, well, they're running in opposite directions, why would that be? It's because one half of the strand is the mirror image of the other half of the strand. So they have to be complementary, but it's like a complementary pair. Each half of the pair is different, yet they have certain chemical properties that make them mate with each other, so that when it's all together, they're a complete complementary set.
Right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact cooppy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring away inside you, copying your DNA with exquisite fidelity.
Isn't that amazing? And it's true. It's absolutely true. every second, every millisecond of every day of waking life that you have, this is going on. And it's been going on. It's probably been being refined either by the creator or by millions of years and eons of evolution into the complex creatures that we are. There are simpler ones out there, bacteria, yeast. There are simple ones, and we study those to get a clue of what the more complex versions of it are in us.
It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you.
He blew right by that, but there is a mechanism in our cells, in our nuclei, that is an error correcting mechanism. It looks for flaws. It looks for faults in the copying, and it goes back and it fixes those things. And of course, if we get more errors going on than the rate at which they can be fixed, well, then things happen like aging, disease, other various manifestations of all sorts of metabolic or structural problems that a person could have in their body. Now, what helps this cellular soup work better? Well, a non-toxic environment, good nutrition, all of the right building blocks. When you look at these things operating, this is in real life, this is operating in what looks like a soup, kind of a stew of all sorts of chemicals in our cells. And there's got to be the right mix of all of the supportive building blocks in that soup for this to happen properly. And if you got wrong stuff in there, or if you have like other forms of energy coming in, strong electromagnetics or any kind of a disruptive toxic force, then that'll jangle this process, and it might make things break apart. It might make them form erroneously, couple incorrectly. So there's a whole lot of things that can go wrong, but that's why nature has designed the error correction process in there. Just thought I'd throw that in.
Copied backwards. So this is work from a number of years ago, but what I want to show you next is updated science. It's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding soup of molecules, which I've stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, to regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of its field of view is a single strand of DNA. This huge package of DNA is called a chromosome, and we'll come back to chromosomes in a minute. We're pulling out.
Okay, so I don't need to say it. If you're paying attention, you know what he's talking about. It's really long and it's really thin, and it's got a huge amount of software, genetic code in it. And so for the economy of space, it is coiled up and wrapped up many, many times. Let him go on.
We're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is...
Okay, notice how small it was a minute ago. One of those tiny, tiny, tiny little dots is what we were just looking at. Now you're zooming out to the outside of the nucleus and probably outside the cell.
About a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving.
You're actually seeing big clusters of chromosomes, synonym, gene, synonymous with DNA at one magnification anyway.
The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again,
you've just seen cell division right there. So it makes it basically those things separate And then what happens is they become complete again once the division process is finished, because the other half of the complementary pairs come out of this soup and put these back together into fully formed double helix molecules again.
And you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have two strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really as to how it's achieving that movement. We've been studying this thing called...
Look at all those tiny little dots. Those are coiled up sections of code, information. And if you can get an idea of what the amount of information that can be stored in a chromosome is, given that every one of those tiny, tiny little dots is probably, I don't know, thousands of base pairs of code. And each one of those little dots, all right?
The kinetic core for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that...
Signal broadcasting system. And you know, TMC, we've talked about artificial construction of nanotubules and nano-sized conductors of various types. And I'm not discretely connecting dots together here, but peripherally connecting together the dot that there's communication that happens between this point on chromosomes and other parts of the cell, so that everybody knows what everybody else either wants to do or is about to do.
Everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red, and it's walked away down the microtubules. That is the signal broadcasting system.
That's the communication. That's the communication from that focal point on a chromosome down a microtubule to a destination. He's probably going to show us the destination.
And sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork.
See these little guys walking up and down those microtubules? We have, well, we don't, but scientists have other scientists who are building molecules that do things just like that. They're able to walk up and down, first of all, able to build tubules and microtubules, but also others that are able to walk up and down those things and carry something else with them as a payload.
This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here...
Just beautiful animation. I'm going to let him finish this up, but I'm not trying to propose that everybody figure out and understand every term that he's using here. So anyway, here we go.
That is the signal. It's molecular clock eye candy. We've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier of science, at the frontier of human understanding, is mind-blowing. discovering this stuff is certainly a pleasurable incentive to work in science. But most medical researchers, this is just discovering this stuff is simply steps along the path to the big goals, which are to eradicate disease, to eliminate the suffering and the misery that disease causes, and to lift people out of poverty. Thank you.
Well, that's a very optimistic way of looking at it, and I'll go with that. Yeah. Don't you just marvel at how we have discovered and discerned what some of those inner workings are? Pretty much all I had for the moment here, TMC. Where do you want to go with this?
Well, earlier I found some things that related to nanotech and to genetic therapy. So if I could find the window real quick, then I'll share real fast what I found.
Sounds good. While you're looking for that, interactive comment with our chat group out there. It is this is I almost want to say the final frontier, but then when I use the phrase final frontier, I either go toward the submicroscopic or I go out to the scale of the cosmological. But this is, if we're talking about life in our terrestrial physical world, this is why we're concentrating on this so much, because the development of modified artificial or augmented molecules to alter the function of a living molecule it's intense research going on in this area, and they're really getting somewhere.
Yeah, so I found this. I was kind of striking when I looked at it again. New technique can track drug and gene delivery to cells. This is May 2018. Targeted drug and gene therapies, finding the target cell is only half the battle. Once the agents reach your cell surface, they still have to do something, right?
Right.
Once they get there.
Right.
So they say here that the University of Illinois researchers say they now know how to track and map drug and gene delivery vehicles to evaluate which are most effective at infiltrating cells and getting to their targets. Insight that could guide development of new pharmaceutical agents. The researchers described their new tracking system and finding in Nature Communications, which is a journal publication. It says gene therapies have shown promise in cell cultures and animal studies, but have been less effective in clinical trials. And so they're working on trying to figure out how to make this better. And they're calling this new class of pharmaceuticals, they're calling these things biologics. So gene therapy, AKA biologics.
And you know why it's less effective in vivo applications? Because the biological soup of life is kind of, it looks a little bit chaotic, but it's actually highly organized. But we need to learn more about that organization and get in there with these agents and essentially not disrupt existing processes, which are so elegant and well coordinated. So we need to understand how that stuff works and then build something that goes in and does a very precisely targeted modification.
Yeah, they're saying here the way they're defining biologics different from traditional drugs is that they have to be attached to specialized delivery agents such as nanoparticles or proteins to reach their intended cellular targets. It's interesting, they say nanoparticles or proteins. 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 Now, proteins, what do we know about proteins? What have you heard about proteins lately? Something about maybe a spike protein, but there are many, many proteins.
Yep.
So it says here there's a lack of efficacy, or their lack of efficacy stems from the difficulty in reaching targets within the cells. Obstacles hampering them are poorly understood. So all of the subtle little nuances and the intricacies within the cell is the obstacle at this point. So anyway, here's what they did. They've combined two imaging tools that measure and track how different delivery agents enter the human and mouse cancer cells. Researchers use single molecule imaging, which allows them to observe individual molecules over time, and quantum dots, which act like tiny beacons inside cells. This allows the researchers to see, count, and track all of the delivery agents that entered into the cell. So look at all these little dots, man. They're tracking all the things that they put into a cell, whatever they are, and they're able to look and see where they go. So you can see like the outline here of something that this thing like deposited over here. So think about what they're studying here. They're able to study which materials go where in a cell: ribosome, cytoplasm, into the nucleus.
Yeah, they're putting tags out there so that they can know where, and see where that, if they got a tagged molecule and they watch where it goes and if it gets modified chemically by the cell.
Lots and lots and lots. You could study very, very specific, intimate cellular processes when you are able to do stuff like this. This is one cell, and they're looking at subcellular functions because they're able to tag molecular components or molecules or things, food, for example, just one example,
right,
that a cell would want. And they can track where it goes all the way around in here. It's pretty sophisticated stuff.
If I get ready with your next thing, but if I briefly take the share back, and we talk about, well, I mean, I don't want to, this is the whole show is just for your entertainment only. But if we were to, okay, right here is a manufacturing operation, okay? And what comes out at some point out of this is a built protein enzyme. Enzymes are proteins, by the way, usually bigger ones, or other structure. And in some of what people have heard about in the news is certain kinds of proteins on certain types of virus particles, et cetera. And we don't want to say anything so that, we cause any sort of censorship or anything like that. But what is currently in deployment is something which can go into a cell, and it can take over a part of this process by modifying some of the code on RNA, not DNA, and then spit out a particular kind of protein. And if I run this without running the sound, and you look at this, if you can just imagine that under natural processes, this is just running. It's running. It's running. But if you put other instructions in there and you make it build something else, you basically can get cells to build anything you want them to build. So without specifying and specifically making reference to anything, the technology to cause your cells to build certain proteins which have been designed and coded specifically in that genetic software is something that is now being done. It's being done now. So we're there. We're there. The animation is definitely there. This is, once again, Drew Berry did a really nice job on his animation there, and that link is there. You can watch that to your heart's content. I'll give you back the share, TMC.
Okay. What else did I find while you guys were looking at those videos? I saw the last bit of those videos. Those are beautiful.
Theranostics?
Dude,
Whoa.
look at this. Look at this. So I found this document in my notes.
Wow.
And it's what it is. It's targeted delivery of engineered auditory sensing proteins for ultrasound neuromodulation in the brain.
Okay, translate. Before you go further, let's take that phrase apart. All right.
So targeted delivery of engineered auditory sensing proteins. So an
acoustic sensing.
Engineered auditory sensing protein. So you have a protein that has been designed to specifically modify the way that a cell responds to sound.
The way that it's a brain neuron. So you're going to use acoustics.
Actually, in this case, they're putting it somewhere nearby the ear. But yeah, it's the same general principle.
Yep.
And they're talking about
what's the date on this one?
2020.
Fairly new. Okay.
Yeah. Oh yeah.
Very interesting.
Yeah, especially when you start looking at how microwaves and millimeter waves can create ultrasound in the body.
Exactly. Well, exactly, but always keep in mind there's more than that. If you have more than 1 millimeter wave and they're dialed into particular frequencies, then they can create a third harmonic They can create many harmonics, actually.
Well, yeah, that's the whole point, right? Is that they can induce complex waveforms, or they can create complex waveforms, either
out of as few as 2.
Well, yeah, they can do it before it gets to the body, or they can do it as it mixes with the body.
Right.
And what they're talking about in this document here is sonogenetics, and what that is genetic manipulation of cells to respond to sound, to respond to longitudinal or acoustic waves. And it talks about here being able to genetically modify cells to respond to ultrasound in very, very, very low power, non-invasively. So they don't have to really do much other than transmit ultrasound into you to manipulate cells. So here it is right here. Previous studies have reported that Prestin, which is the protein compound they're talking about, resides in the outer hair cells of the mammalian cochlea, which is part of the ear, and it drives the electromotility, which is just an electromechanical conversion. So the vibrations convert to electrical signals that basically ride nerves up to your brain, and you interpret that as sound. You hear it as sound. Anyway, they go in and they talk about how
that is how we hear, by the way. There's these tiny micro-sized hairs in our inner ears, and when they vibrate, they produce electricity, which goes down the auditory nerve, and our brains have learned to interpret those as every sound that we ever hear, however complex it is.
Yeah, I'm trying to find, here it is. The successful transfection of this Prestin protein, where they injected the genetic therapy, the gene therapy, right, was evaluated using intracerebral expression of green fluorescent protein. So they basically, when they were done with these animals, they diced up the brains and looked at them under a microscope to see how the fluorescent proteins expressed. They were basically used as tags to understand which cells were modified with this gene therapy. And they said in the study here that I want to find.
You don't need to go into the metrics of it too deep.
No. I want to. There's a place here where it indicated that after 7 days, here it is. It says here gene transfection when they did this gene therapy, right?
Yep.
Entertainment purposes only. It's just an example they did on mice with this DNA, RNA, gene therapy stuff to try to modify cells to do something, in this case, like have ultrasound be able to modify the functionality of brain cells.
Right.
And so what they said here was that the first 7 days were great. And it said after 7 days, the gene transfection, the rate at which cells were converting, were showing new expression with this new genetic code, decreased after 7 days. And they said this is consistent with findings from previous studies. So this gene therapy, many studies done, says what I'm extracting out of this with animal studies, says that you have about a 7-day window, basically, where you have rapid gene transfection. And then after that, it kind of tapers off. It says the transient expression might be caused by delivered, in this case, pDNA-induced immunostimulation, blah, 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 blah. I just wanted to share that. Anyway, it says here that what happens here is that
well, hang on. What they're talking about there is that it works for a while, but then the body's immune system comes in and starts to disable it. I think that's what they're saying in that upper part of that paragraph. What's that?
Is that what you're interpreting out of this?
Yeah, that's my interpretation. So the transients of the expression.
Inflammatory response here, eliminate the transfected cells.
Exactly. Yep. Mm-hmm. So, and then of course cytokines come in, and you get the immune response. So the body, what they've run into as a problem is the body sort of throws it back out after a while. But all they have to do,
it says to further prolong the duration of transgene expression, reducing the CGP content or administering antibodies to the cytokines during gene transfection. Administering antibodies to the cytokines during gene transfection
right,
prolongs the duration of gene expression.
Right, exactly. So what they need to do is they need to fool the immune system and divert it to playing with something else so that it doesn't go after these, and make this thing less of a target on the immune system's radar screen. Exactly.
To enable more
long-term,
genetic transfection.
Right. Wow. So that it would be lasting, I mean, ultimately, what they'd like to do would be whatever it is that they're trying to do would be
Deliver
to
the
get
payload, block the immune system, allow the payload to do its thing before the immune system comes back online.
Exactly, and to do it perpetually so that the immune system never goes after these things and gets rid of them. You know, if you're trying to fix somebody's hearing, you don't want the immune system to go in and take apart what you just tried to fix. You want it to stay. So they want it to be perpetually. So therefore, they would need to either design them in such a way that they look utterly natural to the human body, and the immune system would never mount an attack on them, or et cetera, et cetera.
I like that. That's good. So, yeah, it says here, although the duration, you were right, Indy. It says here in the conclusions, although the duration of the transgene expression needs to be prolonged, currently less than 7 days, this approach provides a new perspective for research in brain neuroscience for developing new tools for treating brain disorders such as Parkinson's disease and epilepsy. Very, very interesting paper.
So this does open up my thought about other technologies, biotechnologies that we've featured, whereby they haven't told us that the body kind of goes in and wants to get rid of this stuff, but that probably is active with a whole lot of the technologies that we've featured on previous shows, whereby, yeah, they got it to work, but after a few days, for one reason or another, whatever implant or molecule that they've parked next to a brain neuron, for whatever reason, might get attacked and removed and neutralized. They haven't talked about that, but I'll bet that is a thing, something that they've got to solve. In order to have perpetual piggybacked control of brain neurons with artificially designed molecules, you've got to have them so that they stay there. You don't want the body going in and cleaning those up, right? And putting the brain back to normal. You want them to stay there, right?
Yeah. And then as those cells reproduce and the old ones die off, hopefully have whatever materials are there stay in the scaffolding to work with the newer cells.
Replicate the function, right? Replicate the function as the cells get replaced by new copies.
Maintain functionality during cell replication, during normal cellular life cycle. Yeah. So let's see what else I forgot now. It's been a while.
So that's all right. Think about it. So if you guys know what we're talking about, we're talking about whether it be for better or worse. One of the things that they've got to do is to be able to have these new functionalities be there and stay and be stable and either migrate to the new replacement cell for the old one, or to be integrated into the code of the cell so that they actually get replicated along with the rest of the cell when it's replaced by a new daughter cell.
Right. So this paper is behind a paywall, and I'm not going to pay for it. I'd like to read it, but I'm not going to pay for it. So very interesting. Smart nanomaterials for advanced biomedical applications. Remote control of cellular functions through smart nanomaterial represents a biomanipulation approach with unprecedented potential applications in many fields of medicine, ranging from cancer therapy to tissue engineering. By actively responding to external stimuli, smart nanomaterials act as real nanotransducers, able to mediate and or convert different forms of energy into both physical and chemical cues, fostering specific cell behaviors. A new paradigm is proposed for nanomedicine in order to exploit these intrinsic properties of nanomaterials as active devices rather than passive structural units or carriers for medications. And this is a big deal for pharmaceuticals that we import China.
Well, it's both. I mean, they're, different groups, different labs are working on perfecting the carriers and perfecting exactly what their payload is going to be, and the delivery and the targeting. And then as for what they do, it's not just a payload, but it's something that becomes active, which gets back to what I think it was Chris Voigt that was talking about, where you're actually modifying the logic gate functions that occur in cells by bringing in new stuff.
Yeah, or manipulating voltage potentials across cellular membranes, or blocking chemical receptor sites, or
you could do
or
that.
stimulating chemical receptor sites.
Yep.
Yeah,
or adding a new receptor or adding a new output, so that you've got something else that's looking for a particular output to come out that was not previously an output of one of the cell's metabolic or biochemical processes.
So this part is kind of critical here. These smart nanomaterials act as real nanotransducers to convert different forms of energy into both physical and chemical cues. So what energy are they talking about here? Nanotransducers controlling cells. These little nano pieces of nanotech are inside a cell inside your body. And they're talking about different forms of energy being administered to the body. And then these nanotransducers ultimately converting that energy into some other type of energy that interacts with the physical and chemical cues that go on at the cellular level inside your body.
Well, you remember what I said earlier. One of the parts, part of literally the stock and trade of what goes on in cells is electron transfer. So if you can tap into a little bit of that electron transfer, that's energy. And that can be used to actuate or activate a transducer that has been designed to receive some of the active charged electrons that are flowing in that cell or in that nucleus or in some fluid within a cell to make it energize, to make it activate. And then you're going to emit at a very micro-sized scale, perhaps radio waves, perhaps light, or perhaps some other output function, depending on what they want. Depending on what their detectors are designed to detect, they'll design the particle so that it emits what they're going to be able to receive. Hey, perfect. Look at that. How'd you hit that exact right diagram for what I was bungling my way through trying to explain right there? That was perfect.
Yeah, so there's a couple examples. Magnetic fields, light, changes in pH, more acidic, more basic, or ultrasound or vibrations. And they have genetic manipulation studies for all of these things and nanotransduction materials for all of these things.
Yep.
And it's, anyway.
So what is it that you pick while you got that diagram up there? What is it that you pick? Again, what you choose is what type of output or input lends itself to the part of the biological environment that you're in.
You're missing. So, okay, great. I like your answer. Are you done with your answer? I'm sorry to interrupt you.
No, it's good. Go ahead.
My answer is a little different.
Okay.
My answer is you pick whatever's available.
Yeah.
So from an AI perspective, image the environment, understand what's available, address it, map it, catalog it so it can be referenced and accessed at any time, and do whatever you need to do in the environment. Run the software you need to run. But basically, you come into an environment and you would scan and map and understand what assets are available to you. And what that means is what systems are out there, what individual pieces are out there, what systems interoperate with each other, and what pieces interoperate with those systems that all tie back into the modern electronics that we understand as smart devices that then connect to common central substrates that function as a cloud infrastructure managed with AI. And then you can reach out and use multi-sensor platforms to do many, many, many complex things in the environment, all with AI spread out on top of it. And nano networks, nano-scale networks are now part of the Internet of Things. And we're talking about biological nanoscale networks, ultimately at some point, whether you believe it or not, coming online on the Internet. And that's really what this is all about. When you look at these stimulation techniques, what techniques can be done with something like a laptop or a smartphone? What techniques can be done with something like a modern wearable? If you don't think a laptop or a smartphone or a television can do these things, fine. A modern wearable, something like a headset that you put on that has little electrical pads or, little light projectors, that kind of stuff. How long before these things become commonplace such that they can provide neural stimulation, whether it be through your skin on your wrist or your forearm or your ankle or, you know, the small of your back, or up to and including stuff that they put on your head that provides transcranial stimulation? How long before that's normal? And these nanotechnologies are here. They've been here for 2 decades approximately or more. And off we go with, you know, our modern medical plight that we're all facing with this horrible, you know, pandemic. Ugh. Where's my mask? I need my mask.
Yeah. When we're talking about waves, there's one little thing just to really bend everybody's mind to another level. We're talking about waves, and we have discussed 5G, we've discussed 2.4 gigahertz, we've discussed frequencies up in the terahertz. There is another family of waves which are called, and I don't profess to know a whole lot about this, but they're called scalar waves, and they are a much, much, much shorter wavelength and much higher frequency than most of the waves that we're talking about now. And that's a whole new frontier. That's
not true.
Okay, go ahead.
They don't have to be a much higher frequency.
No, maybe they don't have to be, but they're smaller.
No, they're just, it's kind of like a collapse. I don't know how to, depends who you talk to. The way I understand them is they're almost like a collapsed wave where you're canceling out the E field and you've got the magnetic field there and it functions, but then you ultimately get it to a point where you're creating a standing wave with something. And then that standing wave basically gives you instantaneous connectivity. You can communicate over it. You can transmit energy over it. You can create action at a distance over it with scalar potentials. But it really it's more or less a standing wave of magnetic fields. It's just a magnetic field oscillating magnetic field that's you've got a standing wave there. But I agree. I mean, you say higher frequencies because you're probably thinking Tesla and the Tesla machine and the high voltages and the low current, but the high voltage, so it's safe to be around and all that stuff. So you're thinking higher frequencies, but I don't necessarily personally believe that. But I'm not formally educated, so I could be wrong.
Just a brief. I guess this will be just a brief aside to it. I have a little thing about scalar waves up here that we could go over. And it's just background information.
Go, man. Here.
If we haven't all fried our brains already on learning new stuff, we can just push it a little bit further. Let me put this thing up. It's only a few paragraphs. So what are scalar waves? And let me get this positioned so it's pretty good here. Okay. What are scalar waves? Scalar waves are longitudinal waves, somewhat like sound waves. Unlike transverse waves of electromagnetism, which move up and down perpendicularly in the direction of propagation, longitudinal waves vibrate in line with the direction of propagation. Transverse waves can be observed in water ripples. The ripple moves up and down as the overall wave moves outward. So you throw a pebble in a pond and you see a kind of a wave shape emanating out from where the rock dropped in. But also what's going on at the same time is the water is moving up and down, as well as the wave traveling tangentially away from the thing that you put in there. It's that the water's also moving up and down. So ripples move up and down as the overall waves move outward. So there are two actions. One is up and down, and the other one is propagating in a direction outward. Technically speaking, scalar waves have magnitude, but no direction, since they are the result of two electromagnetic waves that are 180 degrees out of phase with one another. So TMC, what I would submit to you is there's yet another frontier of interaction of electromagnetic waves that could induce a scalar function that we haven't even talked about yet. And and it goes on. This results in a kind of pressure wave. So keeping in mind that we have ways of inducing and generating these types of waves, and it could be that our Some of our energetic cellular and biological processes actually generate scalar waves as well, and we haven't even talked about it. So, and I'll go on to just the last couple paragraphs here. Vibrational or subtle energetic research has helped advance our understanding of scalar waves. One important discovery is there may be many different types of scalar waves, not just electromagnetic. There may be vital scalar waves corresponding to the vital or key body. Emotional scalar waves. Gosh, who knows, guys?
That's chi.
Oh, chi. Thank you.
You're fucking with my chi.
Your chi. My chi is going to try to make amends with your chi. We'll have like chi piece, a piece of chi. Mice like an occasional piece of chi once in a while. Mental scalar waves, causal scalar waves, and so forth. There is so much more to the frontiers of research that when we get knee deep in the animations about how DNA works, there are processes and sub-processes that are most likely operative there that we haven't even got on the map of our animations yet. Now, general properties of scalar waves, and I'll wrap it up with this, that they travel faster than the speed of light. They seem to transcend space and time. Could there be any tie in there with quantum effects? And I just put that out there just for people to imagine and think, because we have proven certain quantum effects do happen. Cause the molecular structure of water or other biological molecules to become reordered. Scalar waves can, none of this is proven, this is theory. This is from, I can put the link to this out there for you guys. I Awake. Positively induce immune function in mammals and are involved in the formation process in nature. Anyway, that's that. I just thought I'd just go off on a little tangent there briefly, and I have a couple of other things I was thinking of. But if you look at how we get from where we are now to being able to build structures like this thing, put this back up real quickly here. Okay, build structures like this thing, we're getting there. And it's done one little step at a time, one little perfection of the material science and making it innocuous to cells' normal processes so that they don't go in and attack it and reject it. And we're kind of working our way there. In previous episodes, we've featured how you can use DNA to build various structures, whether they be sheets, pockets, rods, envelopes. DNA is really a great structural material. It's got two purposes, really. One is it's a really great way to store software or genetic code very compactly, but it's also really good for other things, building these submicroscopic structures. So kind of leaving it at that for the moment. I actually kind of don't know where we're going to go next, but I got a ton of stuff. What are you thinking?
Let me have share. I'm going to take share.
You got it.
Yeah, because there was some other stuff that, since we're on the DNA kick. So here's another one I found earlier on. Genetically targeted magnetic control of the nervous system. And this was a good document to kind of highlight because they talk about a few different things here that we can look at, or at least highlight.
Oh, hang on a second, TMC. Sunshine Country Chickens. I saw your comment out there. I'm not going to read it or put it on airtime because we have certain strong trepidation about mentioning anything like that because it'll wind up getting our video taken down. But note that your comment has been acknowledged, and I encourage everybody out in the chat to go back and forth and discuss that further. Go on, TMC. Sorry about that.
Yeah, no worries. So this document talks about genetic manipulation of a biologic so that it's more responsive to magnetic fields by telling the body to produce more ferritin proteins and have them structure themselves in a certain way to then be able to exercise some type of control using magnetic fields. So they
Right.
talk about optogenetic and chemogenetic actuators, which are other ways to manipulate the nervous system by adding genetic information into a biologic to make it more sensitive, in this case, to light and to chemicals, designer drugs.
I have a little tangent to add into that.
Yeah.
Whenever they integrate a metallic atom into whatever it is that they're building, that opens the door for either magnetism, if it's a ferrous atom, such as iron, or it also opens the door for either the supply or the sucking up of electrons, because metal molecules are really, certain ones are better than others. They're really good at either soaking up or supplying electrons. So if they know what they want to build and they know that they're going to need an electron supply, if they build a molecule in a certain way so that they can use the metal ion almost as an atomic battery, where when it's in a state of charge, it's got electrons that it can supply, or it's going to receive electrons for something else. So keep in mind, at the atomic level, metal ions or atoms are always going to be handy in building some of these things.
Yeah. So anyway, this is they added this genetic material into different scenarios. I read here early on they tried it in a live study and it didn't take, but it took in a petri dish.
That paragraph on the right, electrophysiology in the mammalian brain, I think it was up just a little bit. It's pretty dry and pretty technical stuff, but you know what, everybody, that's what we're here for. We're here to help us all sift through the highly technical and get to what the bottom line is. So here in that paragraph, electrophysiological characterization in a mammalian brain, preliminary experiments prompted them to determine the temporal kinetics of what they're calling Magneto 2.0 activation, because the future utility of Magneto 2.0 is contingent upon rapid activation in response to magnetic fields in living tissues. So I would just connect a couple dots there. When we're talking about living in an environment where there's a whole lot of electromagnetic radiation around us, and it's all running literally at computer speed, it's running real fast, and it can run slow, but it can also run real fast. So if you want to elicit a magnetic response in a tissue, you're going to be looking for a quick responding interface.
Yeah, these are some of these studies that we're showing they're preliminary, but what they do is they show you the trajectory, they show you the trend, they show you the path or the focus. Or focuses of interest. What
they're driving at.
Yeah. And this is very, very, very interesting science.
So now we have, don't go too far beyond that one either. Where'd it go?
It's all right.
That's it. It's up right underneath the thing that says figure 2 in bold. So they apparently they did something to a mouse, and they tried this Magneto 2 on the mouse. And they were looking for, they basically were looking for what effect it had and whether it lasted or not. So I'll just read this. After they did this to the mouse, they then took the mouse apart and they did brain slices to see what happened. And they looked in one very, very specific part of that mouse brain, which is the hippocampus. And for anybody of you who've been following some of our other episodes, and maybe even some other ones on one of our sister channels, the hippocampus is a kind of it's a really important small organ in the brain. So the idea that they were trying to virally transfect some of this Magneto 2 into that part of the brain, that's indicative of a direction of research as well. What does this mean? Well, certain parts of the brain, the pineal gland, the hippocampus particularly is associated with modulating overall behavior of the whole brain. All right, it helps to set some of the priorities of how the rest of the brain works. So that's a central control point for other areas of the brain. That would be a reason why they might want to try to influence the operation of the hippocampus. So in item C, magnetically evoked spike train of a current clamped neuron transduced with magneto 2.0. I won't wade through all that nomenclature because none of us are going to know what it means. Neuron was stimulated with a 50 millitesla static magnetic field.
Yep.
I don't know. Yeah.
Delivered with a permanent magnet.
Delivered with a permanent magnet. Yep. And the graded bar represents the magnetic field experienced by the neurons during the initiation and ceasing of the magnetic stimulation as the magnet was brought toward the brain slice using a micromanipulator. So they got a tiny magnet and a tiny slice of brain, and they're looking at how they can influence the state of those cells in the hippocampus with a magnet.
That's right. Transfect the mice, kill the mice, chop up the brain into slices, experiment with the slices. Basic.
It's interesting that the transfection was successful in getting what they're calling magneto 2 into that part of the brain. I mean, they actually got them to the target. You know, they got them where they wanted them.
Yeah, I need to read this. I want to read this paper again because they could sacrifice the mice, slice the brain, then transfect it, or they could transfect the brain after they've already sacrificed the mouse.
You could do it either way. Ideally, you do it in vivo, you know. But
yeah, I think I read up there higher up that it didn't necessarily take, or there was some issue with that. They made mention that it was a disclosure.
It may have caused a mouse fatality.
I don't know if it was that or it just didn't work. Like they didn't get it to convert the way they wanted it to. Like the immune system was in the way, kind of thing. Now, you know,
the one where they've got the zebrafish. This suggests that the zebrafish was alive. They began validation of their magneto 2.0, and once again, this magneto 2.0 is merely just getting magnetizable and magnetic atoms into a particular part of an organism. That's all they're trying to do here. They first sought to remotely modulate a simple behavior of the zebrafish. They transiently expressed magneto 2 in Rohan beard sensory neurons. I got to look up what kind of neurons those are, but I have a feeling that they're unique to zebrafish. Using regulatory sequences of neurog I promoter. They identified mosaic zebrafish expressing the magneto 2 in those neurons by selecting animals that also expressed a co-injected fluorescent marker. These guys, you know, I mean, I'm just a geek, you know, I'd love to play with this stuff. It's fascinating. These guys working on this and girls working on this stuff in these labs. Wow. Just really cool stuff.
Yeah, this is just what they're telling us about. I mean, this is...
And there's another thing down a little bit lower. Right there. Magneto 2.0 led to an increase in calcium signaling within Rohan beard neurons. Well, at any time that they get something to create an altered process in a cell, then they can go back to the drawing board and say, okay, well, we can get this magneto 2 to change the calcium signaling. Now, how can we exploit that, right? What can we use that for? And once again, we keep going back to previous episodes, but we've talked about calcium channels in cells, and we've discussed how being able to control or speed up some of that calcium channel transport can alter the state of a cell. Hint, hint, a neuron. You can change the charge of a neuron and either get it to fire or not fire based on whether the ion channels are opening or not opening and changing the charge from within the neuron versus the outside environment. So one more potential use of something like magneto 2, but I don't know if that's exactly what they're driving at here. But now we go on to the lower right where it's in bold, says remote control of neural activity in freely behaving mice. Okay, so they wanted to do another experiment, determining whether these ferrous, I'm going to say they're ferrous, I think they are because they're magnetic, are capable of controlling mammalian neural activity in vivo. They performed electrophysiology measurements in free mice, freely behaving mice, and transduced with an AAVI. And you know what that is? That's the virus transfector. You know it? Yeah, that's what it is. That's what it's probably an adenovirus. They're using an adenovirus to transport the Magneto-2 into whatever place they wanted to get it to in the mouse. Okay. Which expresses Magneto-2 in a Cree-dependent manner. I have no idea what Cree dependency is. Please forgive me. They aim to test whether Magneto 2.0 is capable of rapidly activating a large nucleus deep within the brain, which is more challenging when using optical actuators. To this end, they used mice expressing Cree recombinase under control of the dopamine receptor, which expressed in approximately half the medium spiny neurons of the striatum. There's a whole lot of words I would need to look up there. So I'm gonna skip down a little bit. 3 weeks after viral injection, they performed extracellular single-unit recordings with tetrode microdrives. What is a tetrode microdrive? On Magneto-2 expressing striatal cells in freely behaving mice. Now, let's get to the conclusions. They classify, I've skipped down a little bit further, TMC, down to almost the 5th sentence above from the bottom. And go on to the next page, because I want to try to get to their conclusions, because they're talking about their whole experimental process, and I want to get to, okay, yeah, that's it exactly, discussion. Conclusions. In total, they engineered and optimized a genetically encoded magnetogenetic actuator and applied it to the nervous system in freely behaving mice. This is the first demonstration of a bona fide magnetic control of the nervous system using engineered actuators. So they're all about magnetism. They're not about, maybe they'll peripherally work with optics too, but they're not talking chemical, thermal, or anything else. But when you're talking about magnetic, don't forget, that also ties in directly to electromagnetics that can be coming in from the outside. And with dual coincident, slightly alternate electromagnetic signals, you can not only target, but you can control the strength, a precise location of where those two electromagnetics will set up an interference pattern at a specific frequency, at a specific point, in a specific neuron of a specific brain that has gotten this adenovirus-delivered Magneto-2 molecule delivered to it. Anyway, they confirmed electrophysiologically and behaviorally with both zebrafish and mice that they had delivered their encoded magnetogenetic actuator to the target part of the nervous system. I assume it was to a specific part of the nervous system.
Yeah, it was to TRPV4 ion channels. And those are, I think,
you caught that?
Yeah, I think they're mechanosensitive ion channels.
You caught that somewhere. All right.
Yeah, it's been mentioned a couple times.
It's actually good to have four eyes looking at the document at the same time. We can pick more stuff up. So, yeah, what else do they have to say in this fascinating experiment? Oh, yeah, there it is at the bottom, the TRPV4, using TRPV4 because of its small size and known pressure sensitivity.
Yeah, so what they did is they put a genetic code in there to have iron, little pieces of iron, ferritin, deposit right next to these TRPV4 ion channels. So what they did is they put little tabs out there that were magnetic because they were made of iron, ferritin, biologically made material that they can make. Your body makes this, and they can tell your body to make it in concentration and to make it and place it at a particular location. In this case, next to the TRPV4 ion channel that responds to pressure. So what they do is they induce the magnetic field that then acts on the ferritin that's all coagulated or conglomerated together next to the TRPV4 ion channel that responds to pressure. And so when that magnetic field is applied, it actually puts torque, it torques on that ion channel, the TRPV4 ion channel.
Opens it or closes it.
That's right.
Yep. Yep. And if you can control what's going on in ion channels, I'll take us back to a brain neuron. If you can control what's going on in the ion channels, either along the axon or at the core of the brain neuron, you can alter what it's going to do. Basically, I mean, in a word, what you can do is you can change the charge on the neuron and you can cause it to send a firing signal down the axon or not. You could inhibit it as well. This is cutting edge stuff. And you can see why they're working on this. All right. They're working at trying to be able to develop targeted magnets. That's the title sentence right there. That's what they're doing.
Yep.
It's just so cool. Oh, no. This site can't be reached.
Yeah, I forgot what this was. This was it was something.
That might have been a site behind a paywall or something. I don't know.
It's
what
all right.
it was.
It's from biomolecular engineering, new therapeutic outcomes, remote. I don't know what it was, man. It doesn't matter.
New therapeutic. Hey, there it is.
Check this out. This is a different one.
Ooh, this is good. Look at
Wireless
this.
control of cellular functions by activation of novel protein responsive to electromagnetic fields.
This is huge. This is right along the same line. Let me see what the guys in the chat are saying. Yep. Well, we're working on it. Yep. Hope you guys are having fun. This is just right at the core of controlling cellular processes, and it's one of the targets is, well, two of the main targets are always the specific parts of genetic code, and the other one that always seems to come up as a target is neurons, particularly brain neurons. So go.
Okay.
Yep.
So it says they're talking about a glass catfish. This is an animal. It's a fish, a catfish.
Okay.
And it says it's known to respond to electromagnetic fields. So it says here they tested its avoidance behavior in response to static and alternating magnetic field stimulation. Using expression cloning, they identified an electromagnetic perceptive gene. They're calling it EPG, an electromagnetic perceptive gene, from this catfish. That
it's an electromagnetic perceptive. It perceives electromagnetism.
Correct. And so it expresses a protein that perceives electromagnetic fields. That's right.
So you see why you really might want to know what's going on at the DNA level of genes so that you could go in there and make modifications. If they could take that EPG, that electromagnetic perceptive gene, which exists in this catfish, and if they could get that section of code, and if they could bring that in by way of some sort of a viral payload delivery system into human cells, then they would be able to import a new function into the human, where the catfish is extremely electromagnetically sensitive, but the human might not be. They could enhance a human's electromagnetic perceptivity for some purpose.
Yeah, that's what they talk about here. They isolated the gene and they cloned it. They say here they cloned it and expressed it in mammalian cells. It says neuronal cultures in a rat's brain. It showed that the expression of the EPG is confined to the mammalian cell membrane. And they go in and talk about imaging it. And so it says, moreover, wireless magnetic activation of EPG in the rat motor cortex induced motor evoked responses of the forelimb in vivo.
Wow. A living rat.
Yeah, here we report the development of new technology for remote, non-invasive modulation of cell function. So they say here wireless magnetic activation of this EPG, right? It's EPG gene, electromagnetic perceptive gene in the rat's motor cortex, made the rat's
arms wave.
Thank you. Made their limbs move.
Four limbs, forearms. So they've successfully gotten the stimulation at in the location of the brain with electromagnetic input to get the rat to move its muscles in its body. Wow, this is it. Wow, boy.
Yeah. Crazy stuff.
Go back up a little bit. There was it might be nothing, but
what are you looking for? What did you see?
Bioinformatic analysis. No, we didn't need that. Okay. Yeah. Go back down.
Yeah, I wanted to see if here we go. They found this particular type of cell, the HEK293T, in a mammalian cell. So rats were transfected with a lentivirus.
Okay, I was wrong. It wasn't an adenovirus. They're using a lentivirus.
Well, this
okay.
is a different document, just so you know.
Okay, thank you.
So that could, yeah, they probably use more than one type of thing. But anyway, they talk about using a lentivirus to deliver this stuff. Rat culture neurons were transduced with a lentivirus construct containing this gene promoter.
You can skip through that part, but then using calcium imaging. Whoop.
They were, yeah, it's all right. We're kind of just doing this in real time. Wireless activation
of the electroperceptive gene in a rodent model.
Yeah. So
finally, we targeted the rodent limb motor system, which is a well-studied circuit with validated and quantifiable assays for activation. Stereotaxic injection of the virus containing the EPG, the electroperceptive gene, was targeted into layer 5 motor neurons in the right primary motor cortex, that's in the brain, of the area that's associated with the limbs of adult rats. Control rats.
Let's stop there.
Okay.
That's a lot of words to say that they've experimented on rats for a very, very long time, and they are going after a very well-known, highly studied, highly repeatably stimulated neural population that's responsible for regulating motor function in the arms. They're going after this motor function in the brain, but they know exactly what and where they're going. So go ahead.
Right. And it leads me to marvel at how they've gotten it right to where they wanted to get it to in that rat brain. And they did it with viral transfection.
Yeah.
They didn't have to do anything as crude as to take, some sort of a really thin syringe and inject it into that little part. They delivered it with lentiviruses.
Yeah, but maybe they did put it straight through the skull. Maybe they put the lentivirus and they went right into the specific brain region.
Maybe.
Yeah, like localization kind of a thing. Not
but wouldn't
necessarily
it'd be even more elegant if you could design this whole thing and then you just wave it in front of the rat's nose and say, "Hey, rat, sniff this," and then it manages to deliver itself. It manages to migrate itself to the exact right area. That would be a really good way of doing things.
Yeah. So
yeah, we don't know how they got it in there.
They used wireless stimulation.
Well, wait a minute. It says stereotaxic injections. Now, I wonder what a stereotaxic injection is.
Let's find out.
That is how they did it. You're right. They injected it. So the rat didn't pass the sniff. They weren't able to get it that way, or orally, or transcutaneously. They still did have to inject it. Stereotaxic injection. There we go. Stereotaxic injection. Minimally invasive.
Yeah. So this is what
makes a three-dimensional coordinate system to locate tiny targets. The method is used in several types of animal research and is essential to target specific sites in the brain and to directly introduce the brain pharmacological agents that might not otherwise be able to cross the blood-brain barrier. Oh, okay. Simple control of the microinjections is necessary to ensure accurate delivery of microvolumes and using microvolume syringes. Well, that is exactly how they did it. They used a very, very tiny syringe. Okay. That's good. Now we know.
It is. So anyway, those are a couple of documents that I found early on when I was looking through.
I think it could go a lot further in terms of being able to get it to accomplish the proper targeted transfection. If one knows the characteristics of the precise cell type that one is trying to get something to migrate to, then if one learns intimately what the affinities of that cell type are to other man-made molecules, then one just designs that man-made molecule in such a way that it's got no other home but that particular type of cell that you want to get it to. You just design it that way. You know?
Yeah, the word you used, I think, was absolutely appropriate. Elegant. That's
Yeah.
That's what they're after.
Well, cool stuff. Last words on this little odyssey that we've gone on. We would love to back off. You know, we'd love to just back way out and talk about things like 5G antennas and cell phones and things like that, and and out at the very macroscopic scale, looking at electromagnetic wave propagation and data communications and big computers. But the thing that keeps pulling us, it keeps bringing us back to the topic of humans and in this case, laboratory rats and mice, don't forget the mice, is that's what this is all aimed at. This is what so much of this research is for the purpose of, is being able to develop connectivity and influenceability and modulation and control of organisms. We keep getting pushed back to this because that's what it's all about. It's not so remote and removed as you see the big cell tower antenna up on the hill, and then lo and behold, you have this vision of how all those waves go out to everybody's phones, and then everybody can make a phone call. Yeah, there's that too. That also. I mean, that's almost the easy part. This is the super cutting edge stuff. This combined with mapping in extreme detail particular sections of genetic code, whether it be a zebrafish or a yeast cell or a mouse or a human or a monkey, that is where it's going. And then the whole other end of it, which is exploiting the capacity of biomolecules to be turned into logic, that they already do function as logic gates by nature, but to turn them into controlled logic gates, or reprogramming them or augmenting them. That's where this is all going. It keeps heading there. So that's why we're on it. And we keep discovering more stuff. Absolutely amazing. What do you think? Did we
I think it's pretty good. I think we need to figure out what we're going to transition over to next.
Yeah, I agree.
Yeah,
I agree. Well, we do have a couple of areas, and they do sort of, one of the areas does move into that field of action and communication at a distance that seems to disobey the laws of time. And the quantum portion of that, and that other thing that we started some long time ago, which we only grazed the surface of, which was the gateway experience. There's a few other areas we can go in, and I think we'll keep everybody in suspense for a little while. And you never know, during the intervening week between now and when we run again on stream, we may have discovered more that is connected to this and pushes further to, we want to we want to have you right up as best as we can to the state of the art of what's being developed, let alone getting ready to be deployed. So, all right. Well, let's see here. I'm leaving it to you to close out, and I'm just checking in with the chat. And there we go.
Yeah, come back next Saturday. We're going to continue to explore.
All right.
Good night.